G.Sridevi is in her third year of PhD in the GKVK, University of Agriculture Science, Bangalore. She is working on the application of anthropogenic liquid waste, in short urine, for various kinds of plants such as maize and banana, and now with the potted culture, to see the reaction of these plants to this application on their fertility, on their growth and on their fruiting. As you see, she has to do a lot of collection of human urine, and she's doing this from a school. In Dodbalapur, called the Swami Vivekananda Kannada Medium School in a town called Dodbalapur, which is about 35 kilometers from Bangalore. There are sage warnings in the urinal. The urine is picked up in all these cans and has now been brought to Bangalore into the GKVK premises. And Sridevi will explain to us what's happening here, Sridevi.
So primarily treated human urine. First we collected from Ecosan toilet. First primary treatment we did through some aquifer plants by using local aquifer plants. After that, repeated here excellent column studies.
Yeah.
One by third soil. 1/2 sand mixtures. In between coffee filters and Wattman number filter we used. In between that jelly putty for phosphorus toxicities to reduce the phosphorus load. Here a muslin cloth used for fine filtrations.
Right.
After correcting this, you see the difference between
the color of the urine, which is now a clear white
color solution
as it drops out. Whereas when it's up there, it's yellow in color, which is normal urine color. And why is this being done, Sridevi? Why are you doing this filtration?
To reduce the microbial load and reduce the heavy metal toxicity
in the urine. And how are you going to check that?
So frequent interval I am collecting the sample and doing analysis.
Okay. And then after this filtration,
for
what will happen?
pathogen load, I send the sample to Salem Biomedical Research Foundation Department to check whether it contains any harmful pathogen, Leptospira, Salmonella and Shigella. These three microorganisms we are going to identify in sample whether it contaminated or not.
Right. And then what are you going to do with this urine?
After filtering the sample, I will transfer to a greenhouse for irrigating the pots.
Right, so it will go to pots in greenhouse. How many pots are you?
33 pots.
And what will you be growing in the pots?
Radish and French bean.
Radish and French beans. Why radish? It's a root crop, isn't it?
Root crops, yes, sir. Whether root crops will be having any residual effect if use human urine. Already we done maize and plantation crops, we got good response. So we selected for pot culture root crops, whether tuber will have any residual effect or any pathogenic effect. So we are going to conduct a mini study on this.
And what was the response to the crops, say for example maize, to the application of the urine?
A very good response when compared to chemical fertilizer. We achieved 10 kgs of maize extra. For example, chemical fertilizer performs 79.5 quintals. In human urine, 80.5 quintals we achieved. Nearly more or less 10 quintals extra.
Oh, I see. Oh, okay. And banana is now growing very well.
So it shows good response, very good planted, number of leaves also more. It's a healthy plant. It shows some healthy performance when compared to controlled and chemical fertilizer.
Professor Srinivas Murthy is guiding you.
Yes, sir.
And you're doing this as a PhD project.
PhD.
Wonderful, Sridevi. The application of Ecosan, specifically human urine, in various tests to develop ultimately a protocol for the safe handling and safe application of human urine for plant growth and to capture the nutrient contained there.
Frequent applications.
Frequent.
At what time you want to irrigate.
Right. Thank you, Sridevi.